We're on the last session now, so hope you've had a good day so far. We've got two great speakers, and looking towards really when we have electronic health records in every hospital, in every primary care surgery, linked to biological data. That's when we can really practice stratified medicine. And one of the leaders in terms of one of the heads of the FAR Institute is Professor Harry Hemingway from UCL, and he's going to tell us about the vision with regard to stratified medicine and electronic healthcare records. Thank you very much, Harry.
Thank you for that introduction. I'm going to confine my thoughts to JAM today largely, so I'm not going to give you too much of this wonderful future that Munir paints. Another title for this talk could be how do we in the UK gear an entire country for, if you like, the White House Challenge, the new era of data-based and more precise medicine. So what I intend to cover is briefly some thoughts about why we might be interested in scale, a little bit more about what are these data, these electronic health record data, these linked record data, what are they? Are they any good? Are they fit for purpose in our stratified or precise medicine approaches? And then give some examples of how big data may be helping, both in the getting at high resolution phenotypes over time, diseases and biomarkers, and thinking about a translational pathway, the definitive experiment of a randomized trial, the run-up to that, the prequel, and what comes afterwards, the sequel. And I'll end by talking briefly about future directions in UK FAR. So why do we need scale? I'll just give you some examples of why scale matters. So at a molecular level, diseases are rare. So this, an example from cancer, taking across 21 cancers, non-silent mutation frequency. Of course, as you well know, those frequencies are uncommon. And if one wants to do studies picking up cancers with this biomarker, this genetic or indeed other stratifying biomarker, you need large populations. So let's work through some of those numbers. So the cancers in the rows here with those different frequencies of biomarker. And in green, those would be the millions of populations that you would need to access to pick up 250 new cases per year of your cancer of interest. So green, you could do that in London or east of England or Scotland. The middle colour there, the yellow, you would need the population of the UK, and the white, of course, you would need international collaborations. So that's one example. And put that in context, I appreciate this slide is a little bit old, but if you think about the median sample size in pharmacogenetic studies during this period, it was tiny and didn't change. Another reason that we need scale is if you take this from a drug development perspective, if you think we have only something like 1,400 drugs that are prescribed in the UK and the US, and about 14,000 diseases. Most of those are syndromes. And if we want to efficiently accelerate drug discovery, so-called Eroom's law, the palindrome of Moore's law, if you like, the return on investment for bringing new drugs to market, through approaches such as genomic approaches to drug target validation, if we want to efficiently conduct the definitive experiment of the phase 3 trial, and if we want to identify new indications for existing drugs, so-called repurposing, scale matters to all of those. And many would say we are definitely not there yet. It's a journey. So what are these record data we're talking about? So here's some JAM today. So today, in the FAR Institute, we can access 10 to the 7 populations, ages from womb to tomb, all prescribed drugs, all laboratory values, all the diseases, longitudinal record. And of course the cost to the research funder of collecting those data is zero because these data are collected in the NHS. Of course we don't have whole genome sequencing on any other than a tiny, tiny fraction of these individuals. But tooled access to those data exists now. So an example of this isn't a data set, this is an approach to data, is linking multiple sources. So if you imagine that along the top you've got truth, the true experience, if you like your whole phenome sequence over time, how you experience health and disease at multiple events. And then some of that is captured in our, arguably a crown jewel in primary care. Some more of that is captured in structured hospitalization data. Different aspects, more phenotypic detail, for example, is captured in disease registries, and then on to death. We all die, right, and pay taxes. So this is an example of one of those sources. So how good are those data? Given that they are not collected under research conditions, how are these data helping? So a very simplified model to think about whether these data may inform and possibly transform our approaches to the translational pathway. And what I'm interested in is whether we can say these data provide the, if you like, the fundamental scaffold for this research, the high-resolution phenotypes to which you could add the omics. So, high-resolution disease, that's important. So for all those people who think that cardiovascular disease is heart attack and stroke, you will expect to see two colours on the next slide that I'm showing you, which is a cohort of, I think, about 2 million people followed up for about 5 years. So you can't see two colours on this slide. You can't even see predominantly two colours on this slide. This is the relative proportion across age in men and women of about 15 different cardiovascular diseases, fatal and non-fatal, diagnosed in primary and secondary care. So that suggests some resolution in these data. The question, though, is do we trust that resolution? So that relates to how we might validate those data. So here's an example, blood pressure, if you like, the simple kind of poster child example. Blood pressure is related with the incidence of heart attack. Well, we knew that already, so that tells us that this data is able to replicate signal we already knew. What about taking one of those rarest, the smallest cells in that previous slide, abdominal aortic aneurysm? Systolic blood pressure, not related at all. This is one of the largest, I think it is the largest cohort of abdominal aortic aneurysm. So, oh dear, is that a problem? Have we misrecorded abdominal aortic aneurysm? It can't be a problem because diastolic blood pressure is strongly related. So that suggests some mechanistically relevant process in abdominal aortic aneurysm not present in myocardial infarction. So scale plus clinical resolution, in principle, may lead to new insights. Here's another example in an inflammatory biomarker. I'll ask you to put your hand up and say what you think the biomarker is. So I'd also invite you to see that this inflammatory biomarker is strongly associated with some diseases. I don't know if I've got a pointer here. Yeah, I do. But not associated with bleeds in the head. Anybody want to hazard a guess of what the biomarker is? I know what you're going to say, by the way, because I've asked. CRP. Everybody says CRP. But actually, clinically, very few people record CRP. This is the much measured, always ignored neutrophil count, not mechanistically unimportant neutrophil count. And these shaded areas are the normal ranges that every clinician would ignore. So let's get on to some genetics. What about discovery in records and DNA resources? So I want to give you two broad approaches of how we might get at this. And Munir really is at the forefront of this, and I assume that his work will have been showcased before, so I'm going to touch on slightly other areas. So one approach would be to collect biological resources and fit electronic health records to those after the fact. That's a very important approach. UK Biobank, Genomics England, you could argue is doing that. Another approach is to say, start with where the records are and enrich those with biological resources. And that example would be, for example, in Scotland, in Godarts and Share, and East London Genes and Health. And I'll give you some results from those. So the Scottish example in Godarts, one of the earliest genome-wide association studies of drug response, I guess as many of you will know. And the important thing is that these discovery data came from the NHS in Scotland. A more recent example, and I think an important example, one of the very few examples that I'm aware of where English NHS data here linked 99% to related individuals to assess the human knockout hypothesis, one of the few examples of that ending up in a major science journal, showed, and I think extremely importantly and interestingly, that these human knockouts were not associated with health, prescription rate, doctor consultation rate, and even amongst those with Mendelian disease genes in relevant phenotypes. That would be hard to do in many other countries, very hard to do. So why do we want to get more of these resources at scale? An argument, it's not the only argument, is the drug, if you like, left to right approach for drug target validation, from disease through target to drug, but also from the right to left, the repurposing of existing drugs in search of new indications. One example, a standout example in the world, of course, is the Vanderbilt system. And here, the example that they contributed data to was if you like, this gene mimics the action of ezetimibe and was able to show that on coronary disease, and was able to show that rapidly being able to access large samples of people with coronary disease. They could have done this for any disease. They have, I don't know if it's 150,000 or probably more now, DNA samples linked to a rich hospital electronic medical record. And as a demonstration project of preemptive pharmacogenomics in about 10,000 patients, they show that the majority of people have an actionable genotype across these clopidogrel, statin use, warfarin, and other drug-relevant pharmacogenetics. I guess that's very familiar to you. But we need, and we are getting with Munir and others, the next steps from this kind of approach in the UK. And as a plug to the next talk, one relevant array here that's been jointly developed with Sanger EBI and colleagues at UCL is outlined on this slide. So, in a sense, everything I've said so far is sort of background saying, well, the EHRs, they're pretty interesting. They seem, there's quite a lot of signal in these, and would really help us inform our trial design. And just a methodological note here, that when we talk about stratified medicine, there's sort of very broadly two thoughts. So this is a MRC-funded partnership here. Very sort of two broad thoughts here. You can either have interaction on the absolute risk scale, where your treatment effect is constant at different levels of risk, but in terms of your absolute risk, your absolute benefits are greatest at those at highest baseline risk. That's a very common situation in clinical medicine. More of interest in pharmacogenetics is where there's interaction on the relative risk scale. Keeping those ideas clear and separate in one's mind, I think, is extremely important. So, a couple of examples of using records for randomized trials. I would say the UK has got some great examples, but some other countries have got some really, really good examples. So, for example, in Sweden, this is a registry-based randomized trial about thrombus aspiration. Pretty much the whole of the trial, randomization, recruitment, follow-up, was done in records. We've known, of course, in Scotland, actually for more than 20 years, that the follow-up can be done in records. That's important. So, after the trial, what are we going to do in terms of implementing trial results, identifying strata to pick out different treatment options? If you like, it's balancing the knowledge of benefits and harms, applying the results of randomized trials. So one example, if you like, an exquisite example where you've got really bad harms and very clear benefits, is the decision to prolong dual antiplatelet therapy. So what do the records do here? The records are really important because they can provide, without a shadow of doubt, less biased. In fact, I'm tempted to say they're the only show in town in terms of getting an unbiased estimate of baseline risk. There's not a better option. And you can develop a prognostic model for any recorded benefit or harm. And most important of all is that you've got a setting for decision support. You know, why keep a record? You keep a record because it helps healthcare professionals make better decisions, amongst other reasons. So the example here would be, each circle here is an individual, would be you can calculate your benefits and your harms and work out where you are there and whether you're above a decision threshold, treat, no treat. So that is personalized medicine, interaction on the absolute risk scale. No genetics at all here. Let's take a genetic example. So genetic risk scores for predicting the incidence of type 2 diabetes, they exist, they're good, they're quite strong. So for example, in the red curves here, these are people with normal BMI. So that slope, I think you can all see, is much stronger than that slope. But the big effect here, I guess this is no surprise to you, the big effect here is the BMI itself. So, why am I here? Why do I get out of bed? Well, I guess I'm really interested in this, I'm really interested in this, and I'm really interested in this. So here's a collaboration now in the public domain with Google DeepMind and UCL, Royal Free Hospital, which has ingested, I can't remember whether it's 20 or 200 million blood values, but it's a large number of blood values, and is doing things like this, saying what signal is there in these data, and this is one very specific example, that could generate new knowledge which is actionable. So right now, today at the Royal Free, the decision support tools, the prompts to get clinicians to do different things in order to prevent acute kidney injury is happening and is being evaluated. So I'm going to come to the end of this section of the talk before I finish. So cost effectiveness. Whatever we do, Bruce Keogh was talking earlier in the week, the one piece of advice he gives to clinicians and academics is understand the money. That's what he says. So, how do we understand whatever we want to do in pharmacogenetics is cost effective? Again, the health records, I think, have something to contribute. So here is your estimate of risk. It happens to be in stable coronary disease. Again, here are your estimates of risk. So people say at very, very high risk, you would be prepared to pay more than those at low risk for a given relative risk reduction from a trial or a pharmacogenetic variant. These are important for regulatory authorities to have these kinds of, I hate the phrase, real-world data. What it means is, I think, is unselected, unbiased, meaningful baseline absolute risks. And ultimately, why are we trying to do this? Because we want to deliver better outcomes for this, our country, and other countries around the world. And why do the data matter? It's what Lord Kelvin said. If you can't put a number to something, it's hard to have a meaningful debate. And that's why, when we talk about better outcomes, I think we need to make smarter comparisons. Better than what? So, as a British citizen, I'm proud that we're able to show this rather tragic slide. So this is 500,000 people with a heart attack and survival over 30 days. The UK does worse than Sweden. Why should we be proud? It's because we've got the data to answer that question on care and outcomes. Okay, so to finish, what do I think might be useful in a proposed core-funded UK FAR Institute? There are many things, but I just, for the purposes of this audience, wanted to highlight three. That is scale, stepwise ramping up the scale, adding centers, adding regions. Of course, there's not going to be one big database. Of course, we're not going to get to 65 million anytime really soon, but we need to make steps forward. The second is an intelligent EHR facilitated and supported randomized trial program. And third, and for this audience, I guess most important, is the embeddedness of actionable genomic information in electronic health records at scale to drive precision medicine. Thank you for your attention. Oh, I'm going to show you one more slide. So sorry. Which is I'm so sorry, which is a sort of proposed schema of the FAR Institute. How do you create a national institute if it's not around a disease, a discipline, or one question, but it's around the modern 21st opportunities given by data? And here's a current set of thoughts. One needs to develop methods, governed access to data, leadership, crucially skilled people and partnerships, and then build the science. So for today, we've been talking largely about precision medicine, but of course, in terms of the electronic health record, those data lend themselves to public health questions, improving the quality and outcomes of health systems, amongst others. Thank you for your attention.
Thank you very much, Harry. Open for discussion. Any questions? Let me start off while people are thinking about the question. That urea creatinine and Google DeepMind and in hospital mortality, would that be the same for outpatients as well? The patients who are discharged from hospital, and would that kind of ratio still predict outpatient mortality?
That's an extremely good question. I don't know the answer to that. In terms of their decision support tools, they're focusing on acute kidney injury at the moment. But it's a great question. So those, if you like, ambulatory care blood test values, they're available in the primary care record in many situations. So it would be a natural and important extension.
Okay.
I'm a pediatrician. I'm just wondering about the data sharing has been problematic in the past, and obviously I'm more interested in vulnerable, difficult to reach populations. How, of the needs of children or the mentally ill or the very elderly who might have dementia, how do they fit into these systems?
So it's a great question. So because UK primary care, in a sense, to some extent acts as a concentrator and a kind of common portal for data, people with learning disabilities at younger ages and people with dementia are absolutely recorded in these data. A second point would be to say, how do we get at homeless populations, migrant populations, which are of major public health importance? And the work, which recently won a Lancet Prize from Rob Aldridge and others, has shown how it is possible to probabilistically match existing data sources in order to find out new things relevant, for example, to TB screening of migrants. So it's a really important point that you raise, and I think relates in part to if you put data at the centre, then the range of questions you might legitimately answer is broad.
Yeah, hi, Arthur Holden from the States.
Hi.
Over here, yeah. You were referring to electronic health data almost as a monolithic category of data. It happens to be processed electronically. And there is, within this country, obviously been an evolution and continues on in terms of this. Any impact as you see the type of data and the quality of data from going into electronic health records over time? Any impact on the scientific findings to date? And then I have a quick follow-up question.
It's a really good question. So the short answer is there impact on the quality of data? Certainly in terms of completeness, yes, profound. So around the time of the Quality and Outcomes Framework introduction in 2004, the completeness of many really important data items, smoking status, ethnicity, and so on, definitely increased. So that's a good thing, and GPs were paid. In terms of whether scientifically, it always depends on the research question, right? But in terms of the work that I'm aware of in, say, for example, cardiovascular disease, I guess it's the scale precision trade-off. At the scales that we're working on here, everything replicates. We've not had a kind of, oh my word, that's really odd. We know the truth to be X, but we're seeing Y. Doesn't mean to say it can't happen in the future. It's an important question.
And then as you progress with this work, could you see giving feedback either to government or the clinical community in terms of how to improve the quality of this dynamic resource, clinical management, your last chart? Because I would think that would be a critical part of improving this.
So absolutely yes. So medical director of the NHS, Bruce Keogh, that was one of his mantras of cardiac surgery data, was use it and the data will get better and the quality of care will get better. And we're already seeing that, particularly in hospital data. I showed you very, very little about UK hospital, deep UK hospital data, which in places has got a long way to go. But feeding it back to clinicians, making it actionable, making it relevant to the financial bottom line of trust boards is one way of doing that.
Please.
I know you have had some public meetings at the FAR. I wasn't there, but could you tell us a little bit more about what you're doing to reassure the public about the ethical use and collection and use of their data? I know that there are a lot of patients who are very keen for their data to be used, but that doesn't mean that we give you carte blanche.
So it's an extremely important point. So there's a national program of involving public and patients at really all stages of the research process, right from prioritizing which research questions are focused on through to how the results may be communicated. Secondly, there is I think, very successful Twitter campaign, Data Saves Lives, which has attracted some attention in the public. But I don't think we're doing enough. And I think, furthermore, that I would ask the question of whether we're the right people to do this. I think, because in a sense we're conflicted, right? So I happen to work in a university and I happen to think working on these data are interesting, so I'm going to put that case. I believe it to be true, and of course we take the information governance super seriously and so on. But I think the case is better put by people who have no conflict other than I want the health of myself and my family and the citizens around me to be improved, because right now, Google says 100,000 people die per year because we don't mine the NHS's data. I've got no idea where they got that figure from, but if it's a fraction of that, that is a public scandal, and it should be the public, not academics, that address it.
Okay, thanks very much, Harry, for that. A lot of questions for you. Let's move on to the last talk, if we can. You've heard a lot about Nextgen Sequencing. You know that the 100,000 Genomes project is using Illumina as a key provider for the sequencing, and we've got Dr. Mark Ross from Illumina who's going to tell us about what they're doing with the genomes for medicine and using it as diagnostic tools. Thank you.
Thank you, Munir. So as you can see, my title is a rather general one, and I will start off with some general observations, but the second half of my talk really is devoted to the Cancer Research UK Stratified Medicine programme, and I'm very grateful for the opportunity to present the progress in that programme on behalf of the groups involved. So this model is one that we've been thinking about, along with many others, for some time, and many of its aspects have been referred to during the course of the day. So at the bottom is the idea that as particularly sequencing technology advances, we can consider using genomic information in different clinical settings. So, for example, to look at cases of rare inherited disease, to look at cases of somatic disease, perhaps even to consider monitoring individual patients, cancer patients, over the course of their disease, using either sequencing of the tumour tissue or liquid biopsy information. We've also heard various points that the bottleneck currently in this process is no longer really with the generation of the genetic information from patients, but it lies more in the interpretation of that information, and relieving that bottleneck and moving the bottleneck along, if you like, will require the top part of this diagram to work well. In other words, the interpretation will be critically dependent on gathering together, aggregating information from many patients with the same symptoms with their clinical information. So obviously Illumina is known as a sequencing company, so why are we thinking about this model? Why don't we just think about the sequencing? Our group actually thinks about the whole of this process from the sample to the answer. And when we became involved in stratified medicine for the first time with CRUK, I went to visit David Gonzalez de Castro at the ICR, and he said, you can't just drop a sequencing box in our laboratory and expect us to get on with generating the right kind of information and managing that. We need support for the whole process. So that stuck with me, that comment, over many years. And so we are still heavily focused on making sure that the sequence can be the best it can. So sequencing the most difficult parts of the genome in the least biased possible way, but also equally important to improve variant calling as much as possible, to get the most sensitive and specific variant calling across all variant types, and to annotate that information, moving on into the interpretation, which, as I've said, will be critically reliant on aggregation of many genomes. But also there's the question of the samples, so dealing with clinical samples, which particularly in cancer are very challenging. Now, in the case of cancer, so evolving the technology for cancer medicine, all of those things apply, but in addition, as we've heard again many times today, there are some specific problems. So we're not now looking at a single genome, as in the case of germline disease. We're looking at the tumor and the normal to find somatic variants. We need extra depth of the tumor sequence in order to pick up those low-frequency variants or to allow for sample impurity. The samples, as we saw in Mark's talk, if they've been through the formalin fixation process, which is highly variable between groups, can suffer dramatic loss of AT-rich sequences or dramatic loss of GC-rich sequences. The disease itself is variable and evolves over time and in response to treatment. And we really need, for the variant calling side, we need a lot more samples with well-characterized variants of all different types in order to know how well we're actually doing with the variant calling. So I wanted to just illustrate with a small number of collaborations how we kind of cut our teeth in this area and how we've learned to address not completely, but address many of the boxes on that previous slide. So the first one I'd like to pick out is a project done about 7 or 8 years ago with Mike Stratton and colleagues at the Sanger Institute. And this was really to answer the question, can we take a tumor normal pair and call somatic variants comprehensively? And this Circos plot, in this case, it's a plot showing the somatic variants, about 30,000 in this case, in a malignant melanoma. We moved on quickly from that project to monitoring a patient during the time course of their disease with Anna Shu and colleagues in Oxford. We looked at a number of CLL patients, and I'm going to show you results from one patient. And this was looking, first of all, with whole genome sequencing and then with deep targeted sequencing at the spectrum of mutations that are seen in the patient at different time points during their treatment course. So the initial point is at diagnosis, and this just shows a group of mutations and their frequency in the tumor. And in this case, the mutations don't really vary over the time course of the disease, and the only time they do is when the sample is less pure, so when there's more normal tissue contaminating the sample. But you have other mutations that start off high and then are essentially undetectable in the tumor at the end. Another set which are at low levels to begin with and are very high levels at the end. And there's a final set that you can't detect at the beginning, but become very dominant at the end. With this kind of information, you can work out from these frequencies what the architecture of the tumor looks like, and you can establish certain cellular subpopulations based on whether these mutations are present or absent and their frequencies. And this just shows the different colors here represent those different populations. And what you see is that the red population is dominant in the first two time points when there are very high cell counts. At some point, there's a treatment event that reduces the cell number dramatically, but then there's there's relapse, and a different population is dominating now with a different set of mutations. But critically, even when the patient is in remission at this point, the purple population is still the dominant one that's detected at this stage when the patient's actually in remission. A different project then, a collaboration with Charlie Swanton and colleagues, on a patient with glioblastoma. And really this was just to highlight a couple of things. First of all, this is looking at 3 regions of the tumour from that patient, showing different amplifications across the PDGFRA gene in that patient, which with other amplifications in the patient showed A, heterogeneity, but B, multiple effects on the AKT pathway, which could have indicated particular therapeutic options for the patient. But also this project was a whole genome project turned around in 10 days from the time of receiving the sample to having these results. So this was showing that we could do whole genomes in a clinically relevant kind of time frame. And finally, the project which I'll describe in some more detail going forward. The only reason I've highlighted it here is to remind me to say that in all of these projects, we're thinking about not just whole genomes, but targeted sequencing where that's appropriate. And certainly that was the approach used in both phases of the Cancer Research UK Stratified Medicine Programme. So through these and other collaborations, we've really, these have allowed us to get to the point where we now have these 2 major partnerships that involve large numbers of patients with cancer. The first of those is the Genomics England project, as well as the cancer patients obviously looking at rare genetic disease. I'm not going to talk about that project at all today. But the other one is the partnership with Cancer Research UK and other groups on the Stratified Medicine Programme. Okay, so I've just got one slide on phase 1 of the Stratified Medicine Programme, just to remind you that is completed and the outcomes of that project. So at this stage, the idea was to really evaluate establishing a network to deliver precision medicine to UK cancer patients. And at that time, there were involved the 8 experimental cancer medicine centres, shown in pink. Actually, I think the one in Birmingham's missing from this diagram. And those organisations, together with feeder hospitals sending samples into those 8 ECMCs, were collecting tumours of 6 types. The 3 technology hubs, shown in blue in Cardiff, Birmingham, and at the ICR, Royal Marsden, were using their existing technologies. By that I mean they would use the range of technologies, capillary sequencing, MLPA, FISH analysis, to look at a group of 10 genes across those tumour types. And the funding for that project largely came from CRUK itself, from AstraZeneca, Pfizer, and the government's Technology Strategy Board. And the key outcomes were that over testing 9,000 patients, they collected together the clinical and genetic data, which are now stored for research access. Towards the end of the programme, the technology hubs actually switched to using a next-generation sequencing panel that we helped them to develop. The key part of SMP1, I think, is that it did establish a network of labs, which has now been expanded for the SMP2 programme. So in SMP2, as you'll know, the key aspect of this project now is it's about stratifying lung cancer patients, so non-small cell lung cancer patients being stratified for the population of clinical trials. The whole partnership is overseen by Cancer Research UK and involves Pfizer and AstraZeneca, who, as well as providing funding for the project, are providing agents in clinical trials that the lung cancer patients can go on to as part of the Matrix trial if they have the right kinds of genetic makeup in their tumour. There's an expanded set of experimental cancer medicine centres, which I'll show on the next slide, along with over 50 now feeder hospitals feeding samples into those. The technology hubs, the testing labs within the NHS. The clinical trials unit is situated in Birmingham under Gary Middleton and is arranging the patients onto the clinical trials as appropriate. And our role in this process has been to work with all of these groups to develop a panel that is suitable for the programme. Okay, so as I said, there are now an expanded set of ECMCs shown here schematically throughout the country. The 3 technology hubs are the same as before, so in Cardiff, Birmingham, and at the ICR. And The panel itself contains 28 genes, and the key aspects of the panel and the requirements from CRUK and the other partners were that, first of all, it should work with FFPE biopsy material. It should be a single panel. There wouldn't be any RNA collected, so this single panel had to be able to detect small mutation events, gene fusion events, gains and losses. It would be applied to about 2,000 patients per annum, and the sequencing would be done on a small sequencing platform, the MySeq, not on one of the big sequencing platforms. It would be a tumor normal situation, so tumor samples and blood samples would be collected, and there would be about 2,000 patients per year. So those were the guidelines that we were provided with, and we developed an enrichment panel with that in mind, with those guidelines in mind. The panel itself, as I said, needed to be able to detect small mutation events, such as these two examples in the KRAS gene, and the technology hubs are now confident in calling those events down to a level of about 5% in the tumor samples that they're looking at. But also the panel needed to be able to detect copy number changes, and as you saw in Mark's slide this morning, this is a challenge with many FFP samples because of those gains and losses. And actually it's been, working with Genomics England and with this program has been great to, we're working with Genomics England on how to mitigate those problems where fixation is necessary, so where a fresh frozen route isn't possible. And that will no doubt have an impact on the stratified medicine program where many of the same groups are involved. This just shows an example of a copy number gain in one of the patients. I won't go into the details of this, but this just shows the detection with the same panel of an inversion event involving the RET gene. What we've done here is we've designed enrichment probes to the RET gene shown in blue. We don't design probes to the partners, but these probes close to the junction regions will pull out pieces of fragments of DNA that give you sequence from the fusion point. So it's not necessary to use RNA in this case. Okay, so once the technology hubs have applied this kind of information to the patients, and after the patients relapse from their standard of care therapy, if they have the right kind of genetic changes, they may become eligible for going on to one of the arms of the Matrix trial. As I've said, the novel agents are provided by AstraZeneca and Pfizer and are shown here. And then the various arms of the Matrix trial, which are managed by Gary Middleton's group, the CRUK Clinical Trials Unit in Birmingham. Okay, so just a couple of slides to finish off then on the progress so far in the program, in the SMP2 program. So what we see here are if you look at the three lines, the grey line at the top, that's the cumulative target for samples to be sent by the clinical hubs, those ECMCs, to the technology hubs. The blue line beneath that is the actual sending of samples over time, the cumulative sample distribution from those clinical hubs to the technology hubs. And finally, the red line is the progress in testing those samples at the technology hubs. And what you can see is that the pace of testing at the technology hubs is keeping pace nicely with the sending of the samples from the clinical hubs. So in terms of the absolute numbers, where we are at this stage, so as you can see, just over 1,500 samples have been sent from the CMCs to the technology hubs for testing. Results have been sent back on over 1,300 of those samples, and there's an NGS, a Nextgen Sequencing result on just over half of those. It's about 53%. So what happens to the rest of those instances? So the blue is the other cases, 53%, as I've said, where there is an NGS result. As you can see, 35% of instances, as with the earlier presentation from Mark, there is a QC fail at this stage. There are two major reasons. The first is there isn't enough DNA for many of these samples, so we need a minimum of 50 nanograms of DNA for the library preparation part. So that's one. In some cases, we only get a few nanograms of DNA from the samples. And the second aspect is the purity of the tumour. So the tumour purity has to be at least 20%. And first of all, that's extremely difficult to measure using slides. And the second thing is that in many cases it's difficult to achieve that. So there is a considerable fallout because of impurity in the tumour. And then finally, there are some cases that fail to get the required depth of coverage in the next-gen sequencing. So in deciding whether there's sufficient evidence to call a mutation or critically to call the wild type, as is needed for some genes, there are a set of rules applied on the minimum depth that's required, and that depends on the tumour purity. So if the tumour is fairly impure and then you don't get very deep sequencing across the targets, a sample may fail to have sufficient evidence to make a definitive call. Okay, so just to finish off then, what kind of lessons have we learned from the program so far? Largely here of a technical nature, and how do we move forward on to the next phase of the program? So the first thing, we were clearly aware that there were going to be challenges with the samples, but the challenges are many and varied with these kinds of samples. So the low quantity, as I've already mentioned, very variable quality. Purity determination is very difficult from looking at cells down the microscope. And as I've said, confidently calling wild type for some of the genes in the panel, which is important for some of the arms, can be very challenging. It's become very clear to us during the course of our interactions with the technology hubs in particular that they rely on a simple, rapid and complete workflow. So they need straightforward and rapid library preparation methods. They need a fully automated variant calling system, and they really need automated population of the kind of pre-report that they generate, which currently they take the variant call files and they have a manual process of cutting and pasting from those files into an Excel spreadsheet, and that requires about 70 manual steps and checking by 2 people. It's very slow and it's error prone, and we're working with them on a fully automated system to generate that pre-report, which then has a final intervention before they decide what goes back to the oncologist in the final report. So how do we improve on some of these areas? So we've heard about alternative processing of the tissue. This is important in the Genomics England program, particularly, so the guideline to use fresh frozen wherever possible, but also we're working with Genomics England on how do we get better quality DNA from fixed samples as well. We need methods for generating libraries from lower input of DNA. We need faster and less biased enrichment techniques, and we need a one-click analysis from the sequence to the pre-report stage before that final manual intervention. And I think most importantly, what this has shown to me is the importance of partnerships and networks in this kind of work, not just within the CRUK Stratified Medicine program, but also interactions between programs that have shared challenges being very important. And I'd like to finish there and just acknowledge the various groups and individuals involved in the program. Thank you for your time, and I'll take any questions.
Thank you very much, and for keeping the time as well. Questions? Over there at the end.
This is a really important field you've just described, and it's in the realms of clinical trials. It seems to me that if the novel therapies being developed with your diagnostics as part of that trials get through and are shown to be effective, in future we're going to have to, or somebody like NICE is going to have to parallel approve new diagnostics and the new drug at the same time, because unless this testing and process that you're describing becomes a routine part of NHS practice at the same time the drug is licensed, you're not going to be able to use it in a precision way. And are you looking ahead as to how that might happen and what the costs might be? And of course, there may well be benefits, and we're targeting things better, but it sounds as if the new diagnostics are going to have to go hand in hand with new therapies if they're proved effective here.
It's a good question, but unfortunately I'm not the right person to answer that question. I think in that whole pipeline I described, our areas particularly around making sure we get a good quality sequence data, we call the variants well, and we generate that report well. So I'm sure that people like Gary Middleton are thinking hard about this. But I guess the thing that strikes me about the clinical trials aspect is that the target for populating those trials, when you're armed with the specific genetic information, is actually, I think the target is having 30 patients with that particular genetic makeup onto each of the trials. So that's what strikes me about comparison with a kind of more traditional trials approach where you need very large numbers of individuals with the diagnosis rather than with the more precision medicine information. But it sounds, because of that you're right in your comments.
Over here. So while the microphone's getting there, can I ask a question?
Yeah.
As whole genome sequencing gets cheaper, there will be many providers providing clinical grade genomic sequencing to patients, to the NHS, to any healthcare system. And you'll have read about the FDA, what the FDA are thinking about in terms of regulation of those kind of genomic sequences, but it's very kind of unclear how to regulate that. Can I have your perspective on that, given your experience of the analytic validity and various different approaches you've been through this?
My perspective on
regulation of whole genome sequencing as a diagnostic test.
Yeah, so it's a really key question. I don't have a clear answer, I think. One aspect that I didn't really highlight on the slides is that for the Genomics England project, actually, for the process between the library generation to the annotated sequence, we are in the process of gaining ISO accreditation for that part. And obviously once then there's ISO accreditation for the sample handling at the beginning and for the interpretation at the end, it'll be possible to move away from having to do a validation test. But at the moment, that's where we are with whole genome sequencing. It's not clinically validated for this process. There are clear labs in the United States, as you'll be aware, that have got clear certification now for using whole genome sequence information, and there are strict rules on what they have to do to satisfy the clear protocols on whether they've achieved the right kind of sensitivity and specificity for all different variant types. But it is a big challenge, particularly in cancer. We're thinking a lot about how do we find standards for cancer samples and benchmark the different variant calling algorithms once we have those standards. And it's difficult to get hold of enough of the right kind of samples with all the range of different kinds of mutations that you're interested in to get that, to get that kind of benchmarking. So that's
Okay.
the way we're thinking about it.
All right, thank you. Please.
So you just kind of covered my question a
Sorry.
little bit there anyway, but it's totally fine. So I was curious whether you use a consensus between different variant calling algorithms, or you just use one, or how, because I think they come up with different annotations quite often, right?
They do, and so one of the for looking at germline conditions, what we've done is we have a project called the Platinum Genomes Project, which the data have been freely released and are used externally to Illumina, and that's taking one of the Ceph pedigrees with 3 generations and 17-member pedigree. and it's doing whole genome sequencing on the pedigree, all individuals, and then it's using all the available routinely used variant callers, so not just our own variant caller, as well as different aligners, and then using the inheritance patterns in the family to define what we call a platinum set of variant calls that are inherited correctly through all the members of the family and are called by all of the different variant callers. And that's a kind of what we like to think of as a truth set for those particular germline cases. But it's true to say that different variant callers give you different answers. We measure ourselves usually against GATK, which is the most commonly used variant caller outside Illumina's caller, and we are very close in sensitivity and specificity, but within about 1%, which is still not good enough. We're still about 1% lower, or 0.5% lower on sensitivity than GATK, but that's with a variant caller that calls all types of variants, and GATK only focuses at the moment on small variant events. So many groups prefer to, even though we provide variant calls as part of the package, if you like, many groups still like to realign and recall the data, and that will continue, I'm sure.
Okay, thanks.
Great, thank you very much. Any more questions? If not, thank you very much, Mark.
Thank you.
So I just need to close the conference, so I've just got a few announcements. The first is that we will be sending out feedback forms. Please do send them back to us. It is important to find out exactly how you found the conference, how can we improve the conference for next year. We have packed in a lot of talks here, and maybe there wasn't enough time for discussion, but it was important for you to be able to listen to the kind of state of the art that's going on in this area at the moment. We do hold workshops where there is much more time for discussion and interaction with particular topics in detail, and we have got several workshops already in the pipeline. The first one I showed you was re-engineering of clinical pathways with NHS England. The second one that is coming through June or so, but the dates will be announced very soon and it will be on the website, is with MHRA, looking at regulation of next generation sequencing diagnostics for the NHS. The third one is going to be with the Academy of Medical Sciences on health economics of precision medicine, and there's another one also being developed with the NIH Clinical Research Network Hub in stratified medicine on a particular area, and again that will be advertised very soon. And those workshops, we only have about 40-50 people there, but it does give you time to be able to really interact with people and answer specific questions. We do have some CPD certificates for those of you who asked for those. They are on the front desk, so please collect them. And this is a network. It is free to join. We are quite nice most of the time. And if you do meet with us, we will give you a cup of tea as well. So please join us. Okay, it is free. So now I just want to thank certain people before I close. The first lot of people to thank are the people who really worked hard to organize this for today. First is Christine McNamee, who's the manager for the network, Nathan Elsden, who is the sort of person who's in charge of the website, Eunice Zhang, who's been helping throughout the day, etc., and Karen Chandler-Smith as well from the UK Precision Medicine Catapult. So thank you very much to all of them. So I should also thank the staff at the conference center. This is a great venue. They have looked after us very well, the IT. The food's been great as well, and so thank you very much to the conference center as well. You're going to be sick of clapping after this. Finally, and you don't have to clap right now, but you can clap right at the end, is to the speakers. There have been fantastic, very good talks, superb talks, in fact, and you know, really, really good in terms of state of the science. And there's a lot going on. It's this very exciting area. So please, if you're not already in this area, then to talk to people who are in this area and get linked up. And finally, thank you to you for joining us today and interacting with everybody and networking. And without an audience, obviously, there would be no conference. So you are the most important people. So thank you very much, everybody. Safe journey home.